chemoinformatics and synthetic biology, where we can talk about the concepts of what the bioinformatics is, and later I will give you some introduction on the synthetic biology. And my name is Sergei Shityakov. I'm a professor in the Center of Infochemistry, ITMO University, and I just want to give you some highlights on the Chemoinformatics lab, what we have, what are our goals, and some highlights on the publication, how we are publishing, and what are the resources we have. So this is the Chemoinformatics lab, and usually we have some activities with different universities. Some of them are in Europe and the other in Russia. And this is the cluster, this is the supercomputer, which is in Germany. It's called Julia. And right now we also have a small cluster where we can run some simulations. And we can use it too. So here you can use it too. So those of you who is interested in doing some projects in chemoinformatics, we can also set up computational pipelines here. So this is the Chemoinformatics lab, and the main goals are development of the virtual screening methods, creation of virtual libraries, searching for quantitative structure activity of different substances, and finally we do the experimental validation of different models. Usually we have a lot of experiments, then we try to model the experiments to understand the mechanisms and how we can predict some chemical properties. The models are very important to analyze the chemical substances. Sometimes if we have already pre-trained models, so we can analyze the substances to skip the unnecessary experiments, or to understand what kind of experiments we need to confirm our hypothesis. And this is the highlight of our experiments. They were published a couple of years ago. As you can see, there are some models. For instance, these models describing the protein-ligand interaction. This protein is called blood-brain barrier choline transporter, blood-brain barrier choline transporter. And it's important to transport the substances to the brain, as was depicted here. So you get the blood and you get the brain, and you have the kind of a separation, the blood-brain barrier, and the substances will go through this barrier to the brain. And this is the experiment we did. We tried to cluster different substances, as some of them are permeating the blood-brain barrier as the active substances, as the other are not permeating, so they are inactive substances. So what we have here, we have substances which are active, they're permeating, and the rest is not really permeating, so they are inactive substances. Therefore, this would give us some idea how we can use this transporter to improve the drug permeation to the brain. There are some other projects we have now devised, and here I just put some information on these projects. For instance, the projects which analyze the absorption of antibacterial properties of hydroxyapatite. And yes, right. The treatment, for the antibacterial treatment, applied in the oral surgery, and some projects are organized on the VSG proteins, the proteins isolated from different parasites like Trypanosome brucei. The other project is pharmacokinetics of drug human serum albumin complexes, which we're trying to understand the drug protein interaction, because this protein is important to transfer the drugs to some sites of action, and the binding is important to understand the effect of this. The protein is important to understand the effect of this drug, because if the drug is interacting with this protein, then the activity of this drug is going to change. Some projects are related to cytotoxicity and mutagenicity of different materials like graphene and silica nanoparticles. We also investigate in the different excipients like cyclodextrins, like oligosaccharides, for instance, cyclodextrins. They are important for the formulation of different drug-like molecules. Some substances are closely related to, the topology of the substances is closely related to this excipient molecules like calixarians, or probably in the future we will try some experiments with some other excipients. Therefore, as you can see, chemoinformatics is focusing on different materials. Mostly of them are drug-like molecules, but of course, it's not really committed to the drug-like molecules, but also different biologics like antibodies, antigens, antibodies, proteins, amino acids, different amino acids like peptides, oligopeptides, DNA, RNAs, molecules, aptamers. So, and some hybrids. We can also combine different biologics and drug-like molecules to create some kind of chimeras. This is also possible to model and to do the real experiment. Some methods are related to physical chemistry to understand the interaction of protein-protein ligand interaction, or protein ligand interaction, or protein DNA RNA interactions. So some interactions are modeled using some network analysis like this. And from this analysis, we can see that some proteins are more interacting than the others. And this would also give us some pathways and how we can block this pathway to treat some diseases like a cancer or infectious diseases, the HIV. So this is the HIV study where we're analyzing different proteins related to neuro-HIV. Okay, the definition of chemoinformatics. Chemoinformatics is the generic term that encompasses the design, creation, organization, management, retrieval, analysis, visualization, interpretation, processing of the chemical information, different chemical information. So any possible algorithms that deal with the chemical information might be considered as chemoinformatics tools. The other definition is that chemoinformatics is the mixing of these information resources to transform the data into the knowledge for the intended purposes, making better decision faster, usually committed to the lead identification and optimization. As you can see, the second definition is very closely characterizes the chemoinformatics as the area closely related to the computational pharmacology, where you have the drug lead identification, some kind of a hit molecule, which can and should be optimized in order to be successful in a rational drug design. So if somebody is focusing on the rational drug design, usually the chemoinformatics is the plausible area to start doing the experiments, to start plan and do some experiments. The other definition is the chemoinformatics is the application of informatics methods to solve some chemical problems. Any chemical problems can be solved by, or at least one give it a try to implement some informatics techniques. That means that the chemoinformatics is some kind of application methodology, the application methodology where you could use to improve the experiments, process the data, analyze the data. Actually, whatever analysis is implemented, it can be considered as a chemoinformatics in a very broad term. And the last definition, but not least, that chemoinformatics is a field based on the representation of molecules as objects in a chemical space. One could investigate the molecules in a chemical space to represent the molecules as mathematical entities, kind of using some geometrical representations like graphs or some mathematical strings, algebraic strings like vectors. And you can combine this information, add in some more representations, some different representation, but usually these are two main objects, the graphs and the vectors. And these are very helpful in this analysis. Sometimes this information is very complex, like if you will upscale the molecules in a 3D, you can analyze this is the 1D vectors. The vectors usually smiles and smarts. And the vector, then the graphs are representations of the molecule in a 2D kind of, this is a kind of a 2D information. And you can, of course, combine some information to make it more complex, like a 3D information, some sort of graphs in a 3D in a chemical space. So they're like more close to the reality, like a PDB representation or the different representations of the molecule, SDF representations or MOL2 representations, or they are dealing either with the 3D or 2D graphs. So sometimes even you can add the fourth dimension, some molecular fields to make it even more complex, and so on. So actually, the chemoinformatics is what the molecules are all about, their representations, and their analysis in the chemical space. The chemical space usually is huge, and right now we have only a small fraction of the chemical space, close to some millions of molecules, like 10, 15 millions. Maybe some supercomputers could analyze like maybe 550 or 100 million molecules, but right now it's feasible to analyze a couple of millions of the molecules. So this is the only small fraction of the chemical space we can analyze at the moment. And this chemical space is still growing, is growing fast. So people are using some sophisticated algorithms. Some pharmaceutical companies are generating a lot of molecules in a 3D space to improve the chemoinformatics techniques and to enhance, to expand the diversity, the molecular diversity of chemoinformatics, of drug-like molecules or any molecules you want to try to implement. So the chemoinformatics and the bioinformatics, what is the relation? So I tried to subdivide these two areas into two parts. One is the chemoinformatics, which is here, and the other is the bioinformatics. And what is the difference? Chemoinformatics is more like small molecule-oriented discipline. As you can see here, we have virtual compound library, structure-based design, ligand-based design. Usually it's all about the small molecule, the organic molecule, the small organic, drug-like organic molecule. It's different fragments of the different substructures, different atomic representations of these organic molecules, like shown here. Sometimes you can add different different tools to analyze these organic molecules, like chemogenomics databases to enrich the chemical information. And this is the chemical space, like the universe. It's more like the universe here. So the spectrum of chemoinformatics includes the information, knowledge, rule, number of facts, and the results. So the information you have, usually it's the chemical data representation, the management, and there are some knowledge functions like data analysis, data organization, some descriptors, scoring functions, identification of chemical spaces, some rules. You will add some rules like similarity rules, different cluster analysis rules. You can add some fragmentation rules, partitioning rules, pharmacophore rules, permeation rules. If it's the pharmacokinetics, the absorption rules, and any rules which are implemented to understand the hypothesis. Sometimes people are using these artificial rules. Mostly rules are to make the model more correlate to make the model more suitable for the application purposes, or sometimes it's really hard to understand the complexity of molecular structure. So people are using this kind of rules to make the model more to make the model more in agreement with the experimental data, simply to trying to improve the predictability or the accuracy of the model. So for this, there are various rules which are useful to improve the prediction. And we are also using different rules. For instance, in molecular docking, there are some artificial rules like the torsional rules. If the molecule is rotating, it has a lot of degrees of torsional degrees, which are sometimes very hard to analyze. So people are trying to add some kind of constant, the artificial constant, like 32 torsional angles, where the torsion of the molecule is possible. And by implementing this rule, it's easier to correlate the torsions of the molecule to the experimental data, because sometimes you have very complex molecules with a lot of torsional angles, and these angles sometimes are extremely hard to model, and usually the information provided from this torsional angle is not in agreement with experimental data because of the complexity and the difficulties and intrinsic difficulties in the modeling. Then you get some number of facts, the database mining facts using data mining, literature search, data parsing. Sometimes you are integrating into the artificial neural networks, key nearest neighbor methods. This mostly implemented for the deep learning, machine learning techniques, classification models, and again, these facts are sometimes also very hard to correlate with the experimental data. So there are some kind of a hyperparameter tuning, which is kind of a rule. So you are trying to optimize your model in order to increase the correlation to the experimental data, like a correlation coefficient. And there are some results in terms of the prediction of the biological activity, design of the molecules, retrosynthetic pathways like retrosynthesis, again, permeation. This could be any possible mechanisms you're trying to investigate. And the difference between chemoinformatics and bioinformatics is that bioinformatics is more related, or is more focused on the managing of big data, mostly from the biomedical research. So if the chemoinformatics is more related to molecular data, and the bioinformatics is dealing with the management of big data from the next generation sequencing, some clinical trials, biological molecules like prediction of protein structures, genomic data, different genomic data. See also sequencing as a part of the sequencing data, or just the genomic data like post-translational modifications of the proteins, or some metagenomic profiling, or meta-analysis, and the development of new algorithms and statistics. However, in chemoinformatics, we also have a development of new algorithms. So these are two disciplines also focusing on the development of new pipelines, the new algorithms, development of and implementation of different tools, some software, some chemical software, like graphic user, like some servers for the analysis. And bioinformatics is also focusing on metabolomics, transcriptomics, or any omics discipline which is related to biomedical data might be considered as a bioinformatics. So it's more biological oriented research. Here is more also chemistry oriented research, biochemistry. That's why they are two sometimes overlap. There is some overlap between these two subjects. And what is the pipeline, or the idea of chemoinformatics in a drug design, or just in a chemoinformatics, mostly in a chemoinformatics experiment? What we are trying to achieve in a chemoinformatics experiment is that we have some algorithms, some tools. Usually these tools will help us to analyze the chemical space using some functions. It's called the virtual screening, or the molecular docking, or the high throughput screening, depends on the number of analyzed entities. Here we can screen a lot of molecules, drug-like molecules. The best is, of course, right now is to screen millions of drug-like molecules, but we sometimes limited to our resources, so we are probably analyzing much less, like thousands or hundreds of thousands of some molecules. So it's modest. It's the modest analysis, but of course, sometimes if one could have a experimental database, you don't really need to analyze the millions of the molecules. You can just analyze the molecules which are included in your database, in your experimental database, like we have here. We have the experimental database consists of 15,000 drug-like molecules. And after that, we analyze these molecules to come up to some heat like structures. And for the next step, as a heat optimization and validation. So when you analyze after that, after the analysis of millions of the molecules, you get small library of selected hits to perform the experiment, such as here. Sometimes it's also a good idea to use the robotics. As you can see here, the robotic arm is processing the plates to screen all the substances, and then you can have something like to readouts, and finally you would have a molecule with the best bioactivity. So it's always good to have this kind of cycle, so you can also iterate it to increase the possibility to find this heat molecule. And usually this is heat to lead. When you get heat, then you iterate. It's going to be a lead, then you can iterate, and finally you will get the best molecule with the best bioactivity. So usually you can buy the large libraries of the molecules. You can also buy the proteins. You can buy the virtual libraries, then you will screen these virtual libraries, come up to the best molecule, order this best molecule, do the robotics, and come up to the heat molecule. We also have here the robotics. So those of you who is interested in a robotic liaison between chemoinformatics and the rational drug design. So one could implement this technique to publish, to come up with some very unique substances and publish in the high impact factor journals like Nature and Science. And this can be also kind of optimized and automated without the human interference. Right now, there are some projects people are trying to integrate some kind of robotics protocols, not just for the in vitro experiments, but also for the synthetic pathways, and like Chemify protocols. And here we can also try to do the robotics for the synthesis of some biologics, like like peptides. We already have some algorithms how to design the peptides. If we will add the robotics, then we can probably excel in the rational drug design using biologics. So this is the idea. And for the in vitro experiments, usually you have some small number of the drug-like molecules, so tens up to 100 or 1,000 if you have a very strong robotics facility, up to 1,000, hundreds and thousands of the molecules when you can efficiently screen them in a short period of time. And this could be very efficient and very and relatively fast in terms of the speed and the productivity. So what is the chemoinformatics paradigm? So I already explained in the previous slides that you have the chemical database contains up to 10 in the 9th power molecules. I would say 10 in the 14th power maybe. So then you can just take it and analyze using different these are different functions, different tools like filters, similarity search, structure activity. The structure activity, just a generic term. It also provides the filters and the similarity, and also you can use also pharmacophore and Docken in quantitative structure. So it's kind of generic term. Then you will have some kind of a funnel. Then you will go through this funnel and get some hits, like thousands, up to 1,000 of the molecules. And you can just screen these hits as shown here using robotics by implementing different in vitro experiments. The experiments are usually to check the properties of the substances like cytotoxicity, mutagenicity, permeation, partitioning. Sometimes the partition is important. You will observe the substances where they're accumulating, or for instance, the toxicity is also structured in a different technique, in a different ways, like apoptotic substances, like toxicity in terms of the elevate in terms of the mechanism related to a reactive oxygen species, and sometimes it blocks some proteins of the some vital proteins inside of the cells, like proteins which are important for the protein folding, like heat shock proteins, or proteins related to the protein synthesis, or DNA stability, or the molecular protein trafficking within the complex Golgi, or protein modification, so or activation of different harmful genes within like proto-oncogenes. So it's actually very ubiquitous, very ubiquitous mechanism. And sometimes you will have some misfolded proteins like proteins which are involved in trafficking of ions, like cystic fibrosis, transmembrane conductance regulator, CFTR proteins, which is impaired at some point, and the molecule cannot really the molecule cannot really be effective in the maintenance of the ions, which are extremely important for the homeostasis, for the cellular homeostasis. So the assays, the in vitro experimental assays are very different. Depends on the experimental setup, and usually here we have the MTT as tests, which is the cytotoxicity, so we can actually screen these molecules for the cytotoxic potential. Or you can also screen the substance to interact with some proteins like a human serum albumin as a part of the plasma protein interaction. And sometimes proteins are also there might be the substrates for some transporters which are effluxing, which will influx them and efflux them from the cell, so they will make them less concentrated at the site of action, and the substance is losing its efficiency to interact with the specific receptors, like PGP, for instance. If the PGP is scavenging these molecules, the molecule is losing its biological activity. So one could also screen the molecule in terms of these molecules to be the substrates for this particular transporter, or a BCRP. The PGP, BCRP transporters, they are the same family of the transporters which would exert the substance from the molecule, and therefore reducing the concentration of the drug within the cell. So after the screening, you get some inactives which can be discarded, and at this point you might save the time and money and provide the rational for the drug design. Sometimes you could structure these techniques. The first part, which is shown here, is a ligand-based tools, and the second is the structure-based. I would also add some dynamics techniques, molecular dynamics techniques, different molecular dynamics techniques, molecular docking techniques. And the pharmacophore is a kind of a hybrid. You got on one hand the information about the peptide, on the other you have the information about the receptor, but the receptor is not really present, so it's like a hybrid paradigm. the first techniques are usually very fast, and at this point you can actually screen the chemical database more efficiently in terms of the speed and feasibility. So if you have like millions of molecules, then you can of course implement some very fast filters. The similarity search is also quite fast, and it's possible to screen the millions probably in a couple of days. And then when the similarity is good, then you could probably try to do the different analysis, like a Dawkins analysis, a Mc4 analysis, and so on. So you will add the structure, and actually you will actually see the what would be the biological activity of the ligand. So it's always better to start from the very, very, very fast functions when you get the huge database. Then when you reduce the function, then you can of course reduce the number of the screen molecules up to a couple of thousand molecules. Then you will just try to start from the structure based. Otherwise, it's not really feasible to implement the structure based to a big number of molecules. So far, there are some protocols like genetic protocols based on the genetic algorithms, or the machine learning protocols, some using some machine learning methods like diffusion models, reverse diffusion models, which are quite slow, and especially if we are talking about the diffusion models, you need usually all these machine learning models, they are usually relying on the data, the external databases for the model generation. So right now they're using the expensive CPU, GPU modules to screen the molecules. So the molecular Docker is becoming more sophisticated, and which would of course decrease the speed of the efficiency in terms of the speed of this technique. So this is the kind of artificial, not really artificial, but more like practical terms to discriminate the fast chemoinformatics methods and the slow chemoinformatics methods. You could also see that these fast chemoinformatics methods are dealing with the ligand, and the slow chemoinformatics methods are dealing with the complexes, usually the whole complexes analyzed. Sometimes you get something like protein informations, like if you would probably analyze the proteins, it's also possible to understand the information about the ligand binding, just to analyze the protein by investigating the protein ligand binding sites, and what is the geometry of the protein binding sites, and how these binding sites located within the protein, or they are more like solvent accessible protein binding sites, or what is the number of dissolution penalty for these binding sites, or whether the water is present in the protein binding site to be kind of conduit to improve the binding, to improve the ligand binding. And this might be also the technique, the chemoinformatics technique to investigate the protein ligand interaction. But of course, it's more crippled by the information, by the ligand information absence, by the ligand absence from the whole picture. So sometimes people are focusing on the different barriers, biological barriers, to implement these functions to the chemoinformatics. This is one of the barriers. One of these barriers is the blood-brain barrier. You got this barrier in the brain, which separates the blood and the brain, and the substances are not really permeating through this barrier. Like only 2% of drugs is able to permeate, and the rest is not permeating. And this barrier consists of the endothelial cells, which are located on the basement membrane, and the cells are attached to each other by tight junctions, by different proteins. They're called tight junctions. There are also some other proteins like occludin, claudin proteins. So this barrier is quite complex. There is also a glycocalyx, like glycoproteins are covering the cells, so it creates some kind of halo around the cell, so it makes the permeation quite complex. There are different routes of this permeation, and it's extremely important if some people are analyzing the drugs which are meant to treat some neurodegenerative diseases like Alzheimer's disease. Okay, so can we do you need a break? Because we can have a break and we will continue. What do you think? Hello. I cannot hear anybody. So I don't know whether you want to have a break. You can just let me know. Okay, so it seems like you don't want to have a break. Okay, let's just start with lecture. So then we can just finish probably at 12, right? 12, something like that. Okay, so in my research, I was analyzing this blood-brain barrier by using different experimental protocols like transfer experimental protocols, transfer system, where we had the semi-permeable membrane, and this membrane is mimicking the basement membrane. Then I put some cells on this membrane. Usually this membrane is made of the PET polymer, and it has the same pores which are present in the basement membrane. Then you have to harvest the cells in this membrane, so you will artificially create some kind of a blood-brain barrier which separates the two compartments. One is the apical compartment and the other is the basolateral compartment. which represent the blood and the brain, and you could start doing the experiments with the separation. So it's also possible to model this system here. I think if you have this membrane, but there are already pre-ordered kits, and this kit can be purchased, and you could actually implement this technique to investigate the permeation. We also checking the tightness of the blood-brain barrier using the electric measurements to understand how the mature is the blood-brain barrier. We are calling it transendothelial resistance. And this transendothelial resistance is the increase in the resistance means that the barrier is tight, and if you see the decrease in the resistance, it means the barrier is compromised. And sometimes when the person experiences some pathological changes, either in the brain or in the blood system or in some other organs, sometimes it would affect the tightness of the barrier, which makes the barriers more compromised. So we try to implement some other systems like formulation systems, as you can see here, using cyclodextrin. So one experiment was also done using very lipophilic substances like carbon nanotubes. Either you could use the single-walled carbon nanotubes or multi-walled carbon nanotubes, and the libraries of drug-like molecules, and you can try to screen them through the funnel. And the experiments would be in silico, in vitro, in vivo, ex vivo. Ex vivo is the last one where you can harvest the organs and see how the drug is behaving when you implement these organs for permeation, for instance, or for hepatotoxicity, or for the accumulation of the drug, or for some pathological changes which the drug is exerting to this particular organ. Or the in vivo sometimes is the good model to understand some very complex diseases, because, for instance, a tumor, when you can just take a tumor and analyze it in a petri dish, or we are also modeling. We were trying to create some kind of artificial organs that it could be also considered as ex vivo. We were trying to implement the dynamic blood-brain barrier. We call them biovascs. When we had the intestine, we removed the epithelium from the internal lining as an internal lining, and we have some kind of connective tissue scaffold, and then we cover this biovasc system with the endothelial cells, adding some kind of dynamic properties to the system using a shearing stress and the blood flow to the system. And we were trying to analyze the permeation, trying to mimic the blood-brain barrier in more details. So it might be also considered ex vivo, but it's extremely difficult. You need a special equipment for that. You need also some kind of robotic system to maintain all these parameters automatically. And the readout system. So in some private pharmaceutical companies like Fraunhofer Institute in Germany, the private institute in Germany, they're trying to create this kind of tissue engineering method. Some people also, they're getting crazy about the hard-on-chip and some other stuff. So it can be also considered as an ex vivo technique, or brain on chip, the heart on chip, I don't know, the liver on chip, the cancer on chip, the kidney on chip, whatever cells you are using, you can call it. But the problem is how it's really representing the organ structure, because the organ structure is very complex. The morphology is very, very complex. The cellular arrangements, it has a particular cellular arrangement. So it's extremely hard to recreate this kind of interactions, and you have a lot of inducing molecules that come in from the body. So it's not just like you just take some cells, cover with the silicon. You get silicon buffer. You just put the cells. They are growing. You can create some kind of system which corresponds to the relatively small, the relatively simple system like a blood-brain barrier. Even though the blood-brain barrier very simplified, but for the blood-brain barrier, you just need, of course, you don't have the parasites. Here, we didn't really have the parasites. We didn't really have the glial cells. We just had the membrane, and we had the endothelial cells, and that's it. So we didn't really perform very sophisticated blood-brain barrier. Even for the blood-brain barrier, it's extremely hard, and people just messing around with some concepts, trying to recreate some. They just, for the publicity and for the mostly done for the publicity, although most of these kind of ex vivo methods are not really applicable in a wide range of the rational drug design projects, because they're very approximate. They're not really very precise. They're lacking a lot of anatomical and histological aspects of the human tissue. Even the human tissue is not the organ. So, I mean, right now it's I don't know whether to create the human tissues is not really very easy process, and they're trying to call it like a heart on chip. So what kind of heart do they have on the chip? It's just more like the commercial move to attract some investment, to make people think that you get something incredibly useful, but so far just the best modeling system is the real animal, like a mouse or a rat. Then you can just inject and see how it's really working, and or you can just take the organs and take the heart and try to analyze the organs, how the substance is really what is the impact of the substances on this organ. So right now the in vivo experiment is the best. So you cannot really substitute the ex vivo experiment with the in vivo experiments. And then, of course, there are different in vivo experiments. You can try to use the animals which are genetically very close to humans, and this would even give you the more precise information on the drug effect. So the chemoinformatics, what is the chemoinformatics link to the experimental data and how they are really interacting with each other in terms of the data, information, and the knowledge. So here you can see here's a kind of a triangle. You got a base, which is the data, and you have to analyze this data by using some kind of external information, and maybe you will also get the new information. Most likely you will have the new information coming from this analysis. And then you got the knowledge, which is very important to do the generalization of this mechanism. Some kind of sometimes you can do the generalization, sometimes you cannot. For instance, if you have some proteins, you get molecular docking experiment. When you get the protein dimer, then you will do the docking. Then you will have the information concerning the interaction of some ligands with this dimer protein. So you cannot really generalize, and you get the new information, you get a new knowledge, but you cannot really generalize this information on all the proteins, because some proteins are very different, very similar, but most of the proteins are very different. And the homologous proteins maybe are following this pathway, but some proteins with a very different structure are not really suitable to be taken as proteins for this analysis using similar model or similar pipeline, because their structures is different, and their structure is different, and therefore the interaction site is different, the behavior is different, the stability is different, which will probably give you most likely the wrong result. At this point, sometimes knowledge is related to this specific system. So it can be that you can generalize, but most of the time, sometimes the particular knowledge is only suitable for this particular system. And you can also have a different approach. It's called the inducive learning. from the data to the knowledge. And sometimes you have the deducive learning, which is from the knowledge to the data. So you get some knowledge, some idea, like the click reaction. So you will have a practice with Anastasia, practical. You will have a practical courses with Anastasia, and she will teach you how to do the click reaction. Then the click reaction is something you wanna implement for the drug design, for instance. Yeah, and the click reaction is the knowledge. The mechanism of the click reaction is the knowledge you have, and then you will have a algorithm to produce a big data here, data for the big data. So you might try to use the deducive learning coming from the knowledge as a click reaction to the big data as the database generation. And then you get also the information in between, or information from the data processing. The information from between is the information how to program the click reaction, and the analysis of this click reaction, the processing of this knowledge and things like that. Yeah. Therefore, one could summarize that chemoinformatics learns from the experimental data, because the experimental data cannot be learned from the chemoinformatics. It can be generated from the chemoinformatics in terms of if you are using the prediction. Then you can, of course, try to generate the experimental data. Like we have some projects where we are trying to design the new molecules against some receptors to inhibit or as inducers or inhibitors of these receptors. So we want to do some chemoinformatics if we have the experimental data. So we will create some tools, chemoinformatics tools, to create the new drugs, right? And when we get these new tools, the chemoinformatics tools, we can generate the experimental data. So on one hand, the experimental data is something that chemoinformatics trying to harvest, like the reference. Usually the reference in the chemoinformatics is coming from the experiment. And when you get the experimental data, then you optimize the chemoinformatics algorithms and produce the experimental data. So, but of course, the experimental data cannot be learned from the chemoinformatics in the first place. Like chemoinformatics learns from the experimental data. So experimental data usually is the vital importance to validate the chemoinformatics tools. So sometimes it's possible to generate the new tools, but again, there is some knowledge of experimental data is required, like the information on the mechanism. Then this mechanism was proposed through the experimental data, or at least some understanding of the mechanism. Some investigation was done by using the experiment. Without the experimental data, it sometimes doesn't make any sense. Just some tools, but it's like it's like methodology. It's more like a methodology. Some people can create some pipeline, but what is the practical usage of the pipeline is there is nothing. There is one cannot say anything about the application of this specific chemoinformatics pipeline without the experimental data involved. So, and then again, this is the kind of information we have. We have a data, and then we can come up to the knowledge. So it's kind of deducive mechanism. So we have data, we have the measurements, and we have a knowledge. This is the knowledge where we have different clustering. So this is nicely describes the deducive learning of the chemoinformatics paradigm from data to knowledge. From data to knowledge. And of course, this experiment also learns from the experiment. This chemoinformatics theoretical experiment learns from the real experimental data. Okay, so what is the basic concept of the chemoinformatics? Chemoinformatics is based on the different concepts. One is the quantum chemistry, wave and particle dualism. And the other is force field molecular modeling. And mostly we're using classical mechanics. And the classical mechanics is the first technique you can use in the molecular dynamics, and the quantum chemistry is the DFT, and chemoinformatics is the chemical space. So actually all these techniques these two techniques can be integrated in the chemoinformatics. When you get the chemical space, you process it, then you process it with the classical mechanics, and finally you can process it with the wave particle dualism as a part of the quantum chemistry. Okay. So the quantum chemistry is usually is more laborious and less applicable if you are talking about the structure-based chemoinformatics techniques, because we are using the receptors, different biological molecules. They're quite precise, but in terms of the small molecules, only in terms of the small molecules. If you would like to implement the big molecules, sometimes it's not really feasible to use a big number of atoms. It's usually limited to some hundreds of couple of hundreds of atoms, not thousands, not hundreds of thousands or tens of thousands of atoms. So it's very limited. And here you're analyzing some electronic properties, new electrons and nuclei. So you will have various electronic descriptors, the information, speed on molecular on atomic spins, atomic charges, different atomic behavior. You can also calculate the interactions. You can calculate the the models, the salvation models using these electronic properties. The optimization of the molecules is almost done. The minimization and optimization of molecules is usually done using the quantum chemistry. And the force field, but the problem is that if you want to use at least for chemoinformatics, if you want to use the quantum chemistry, sometimes the molecules are rotating, they're producing a lot of numbers of conformers. These conformers are usually counted in hundreds, sometimes in thousands, and one could not really investigate all these conformers using the quantum chemistry. So the quantum chemistry usually is focusing on the a single state atomic structure. Then you can optimize it, but you don't really know whether it's a right conformer you want to analyze. So it cannot generate the conformation. It cannot generate the conformers. Every conformer should be analyzed separately, and this would also impose a big limitation on the quantum chemistry algorithms. The next is force field molecular modeling. You got atoms and bonds here, and then the idea is to generate the different conformers like a conformational sampling. And the conformational sampling is good, but we are missing the information on the electrons and nuclei because we are treating the only atoms and the bonds, and we submitting the rotations, the translations, the rotations, the movements, different movements, and we're just assuming that if we will neglect the information on the electronic structure, we can probably be successful to get the right information on the binding properties of the molecule. Sometimes this is not the case. Therefore, this technique is mostly implemented as a conformer generation technique. And people are trying to add some sophisticated functions to add in more force field parameters to analyze also the energetic profile of the molecules. But of course, it is less accurate than the first technique. And the chemoinformatics is dealing with molecular graphs and descriptor vectors, which is purely the representation. So you can calculate the empiric descriptors from that very fast, sometimes very efficient, but of course the information on the atomic charges and the bonds and the protonation states and the electrons and nucleus is not really implemented in this representations, only the number of atoms and sometimes stoichiometry. The stoichiometry of the molecule can be also described, but again, the problem with the stoichiometry, if you convert into different keys, the different keys like smarts, for instance, or smiles, yeah, Then of course just the binary keys, then the information is also lost. So sometimes when you're losing this information, you cannot really convert it back and forth. So this only in one direction. And when you do it, then you are losing this information, and the sample you cannot really derive the information on the electronic properties, and of course you cannot really proceed with the conformational sampling of the molecules like we have here. So all these techniques, they have different drawbacks and weaknesses, and one could also combine them to achieve better accuracy. Therefore, chemoinformatics is the field based on the representation of molecules as the objects, graphs, and vectors in a chemical space. So what kind of chemical space and descriptors we have? The chemical space could be represented, as you can see here, graph-based and descriptor-based. The graph-based, when you draw the graph, you draw the atoms and bonds. Usually it's some kind of a picture representing the molecular formula. And you will analyze these pictures. On the other hand, the descriptor-based representations are, you got different properties, molecular properties like this, hydrophobia, electronic, steric properties. These are various properties like right now we get like 5,000 up to 5,000, even more, descriptor-based. The different descriptors, including graph-based and descriptor-based, and you can add your own descriptors like we did. We added the descriptors related to the Voronoi entropy, for instance. This is a new descriptor which we added to the descriptor list. And we could actually structure the chemical space by analyzing the objects plus matrix. Okay, the chemical space, as I already mentioned, it's like a universe. And when you structure the universe, you will get the kind of a solutions where your molecule is located, and what are the other molecules are interacting, or what are the other proteins, and what is the chemical pathway, and all other questions when you're trying to structure the chemical space. So the virtual screening is to find the right molecule. Again, what is the virtual screening? The virtual screening is the method in chemoinformatics which is important to analyze all this chemical space and to find the right molecule. And you get like the needle in a haystack. And which is, of course, the laborious task. You need a lot of optimization, validation, and quality controls, negative controls, positive controls to get this done properly. Okay, so the virtual screening, and it's really very hard to get the right molecule when you do the chemical screening. And therefore you have to optimize the drugs, the they optimize the chemoinformatics methods to come up with a unique molecule like here. So how you can optimize the chemoinformatics and why the chemoinformatics are useful to find the right molecule is that you have number of different descriptor vectors, up to 5,000. You have to use the right ones, the right ones, and this descriptor should be related to processes which you are trying to implement for this analysis. Like if this is a blood-brain barrier interaction, then very important to use the polar surface area descriptors and lipophilicity descriptors like log P, for instance. If this is the descriptor of the affinity descriptor, then I would use the number of torsions and the number of saturated and not unsaturated bonds, because the number of unsaturated bonds means that you decrease in the conformational space here. Sometimes, if this is the toxicity experiment, then I would concentrate on the toxicity related atoms like heavy atoms, or the atoms, the metal atoms, which might also increase the toxicity, and so on. If it's some kind of a hybrid, then probably you have to figure it out. What is the molecular substructure is important for changes, for instance, or what kind of substructure, what is the SCAP descriptors which are using to investigate the scaffolds of the molecule, and so on. So it's probably like this substructural fragments, how many substructural fragments you have for to have some information on the pharmacophore like properties of the descriptor of the molecule. Also, the molecular volume. This is the very important descriptor. If you know that this particular molecule occupies the specific binding site, so you might want to try to match the molecular volume of the molecule, of the ligand, and the molecular volume of the binding site. The charges are also important. The atomic charges as a descriptors, and how the molecule is partitioned, and how the molecule is going through the lipid membranes, and how the molecule is charged in a different solvents, and how you can use them to optimize the solubility of the molecule. There are various different descriptors at this point. Therefore, the descriptors are not really constants, but they are values you have to make sure there is a high probability that your model will be successful. Otherwise, if you will choose wrong descriptors, you will probably come up with the false result, and your model will not really represent the experimental data at this point. So here you can see we got the molecular graph in this molecular graph, there are some descriptors derived, so then we can structure these descriptors. And when you analyze various descriptors, you will get some information like a fingerprint. So it's a sum of the descriptors, the descriptors to produce the uniqueness of the particular molecule. And from that, you will get the model, the QSAR model from different fingerprints. Some descriptors are also efficient as a scaffold tree descriptors. When you get accessory, which as a scaffold, the scaffold is the core of the structure. Usually, the scaffold is the fragment of the molecule, which is present in all the molecules in a database, like in an accessory ring, in a scaffold tree experiment, as shown here. Yeah. So all these molecules, they have this accessory ring, and by implementing this scaffold tree, one could structure this database and see, for instance, how I would just add the similarity function, I would have some kind of scoring function to generate the molecules less similar or similar to this particular scaffold. Then I would get more information on the synthetic possibilities, and on the modification, if I will apply the scaffold hopping techniques, where I have to apply the scaffold hopping techniques. What are the bonds which are important for the molecular optimization, and so on. This might be also important for the toxicity reduction, if we know that some substructures are less toxic or some substructures are more toxic. So switching on and switching off different substructures might also give us the tool to leverage the cytotoxicity. So molecular scaffold describes the core structure of the compound or series of the compounds, and the position of chemical variation in the core structure carrying different substituents like functional groups in different analogs. As shown here, we have different analogs, we have different functional groups, we have similar molecular scaffold, and all this is very important to screen the molecular space. As an example, I would give you the information on this studies when this was published a couple of years ago. And here we have benzotriazole core, benzotriazole molecules. They are known to be the corrosion inhibitors, and if we're trying to align them, as you can see, they are very well aligned. So the alignment is less than 0.5 angstrom here, and this model is based on the experimental data. So when we were trying to structure the corrosion inhibition like this, and with the lipophilicity descriptors like log P descriptor. This is the atomic log P descriptors, means that the partitioning coefficient is the information in which phase the molecule is going into the hydrophilic phase and the lipophilic phase. And on this regression plot, you can see that substances, if they are highly active against the corrosion, they are also very lipophilic. And those who are less active against the corrosion, they're less lipophilic. Then we try to use this is also the correlation with the electronic structure like HOMA-LUMA gap, and the HOMA-LUMA gap correlates with the lipophilicity index as well. Here you can see that if you calculate the HOMA-LUMA gap of the substances, then the highly lipophilic substances have a very high HOMA-LUMA gap. And the HOMA-LUMA gap also characterizes the activity of the molecules. It's especially important for the transistor experiment, when you get the electron movements within the transistor, that the HOMA-LUMA gap describes this event in a transistor. And therefore, calculating this descriptor for the transistor would also be beneficial to improve the transistor activity, in terms of the effectiveness. So we try to combine the information derived from the experimental inhibition of our substances, and we were trying to integrate this information into the corrosion, to predict the corrosion inhibition as predicted. Corrosion inhibition, and we were actually successful, because here what is the idea, how we gotta do it. So simply by using the equation derived from this equation derived from the regression model. So we took this equation to calculate the corrosion inhibition for these particular substances, representing the molecules from the Arabic gum and their activity against the metal corrosion. As you can see, we were able to produce the second quantitative structure activity based on the prediction, the corrosion prediction coefficient, predicted corrosion inhibition coefficient, and the lipophilicity index. Of course, this quantitative structure is less precise in terms of the lack of the experimental data, but at least it will give us the idea that the substances which are highly lipophilic, they are also most likely represent the high corrosion activity, anti-corrosion activity, like the substances you can see here: arabinose, galactose, rhamnose saccharides, and some substances with a very high lipophilicity index will give us the very low corrosion activity. So this was already published. So this is the example how you can actually combine different information derived from the experimental results and integrating into the quantitative structure activity to have a educated guess on what is happening with the corrosion. Why these substances are more active than the others in terms of the corrosion activity. So Just a couple of words on the other paradigm similarity, similarity in terms of the pharmacophore screening. Pharmacophore also is a kind of a similarity. It's more like geometric similarity more than the structural similarity. The structural similarity we have is the Tanimoto similarity. I will talk about it in the next lecture, but here I will just give you some very rough introduction on what we have here and what kind of idea you have from these molecules. So if you notice that these molecules have different structures, here you have a peptide, and here you have the drug, the tirofiban drug, and here you have a oligopeptide, and how you can compare these structures, which are very diverse in terms of the chemical structure, but probably the activity is similar. So why the activity is similar? This would be a good question. And pharmacophore is the method of choice to investigate the activity of these molecules, which are not really similar in terms of the structure. So here we implement in the pharmacophore as a concept of different centers. For instance, you have positively charged centers like amine group, and the hydroxyl group is a negatively charged, hydroxyl group, they are negatively charged. And you got also the lipophilic areas represented by the benzyl groups, benzyl moieties. And if you will analyze these molecules, you can see that the similarity, there is a similarity in terms of the different substructures bearing the same number of the same representation of the charged centers and lipophilic groups. So that information can be computationally analyzed like this. You have a positive charged hydroxyl donors, negatively charged hydrogen acceptors, hydrogen donors, hydrogen acceptors, and hydrophobic interactions represented by the lipophilic areas. And when you would create some kind of geometrical representation of this pharmacophore, then you can see that if you will measure the center of these groups, moieties, then you will get very similar distances from these centers. So that is why the activity is also very similar, because when you will also integrate it into the protein ligand binding site, you can see that these areas are located in the same parts of the binding packet, and probably the amino acid residues are interacting exactly where the positively charged and negatively charged residues are located. So this would be interesting to observe. You can apply this pharmacophore to various receptors, various ligands, and you will get more. You will get more tools to screen the database. You will screen the tools on the positive charges, negative charges, hydrophobic interactions on the particular distances. There are different algorithms also how to process this kind of information. There are more algorithm. There are also ligand scout algorithm. There are Schrödinger algorithm for pharmacophore model. There are some open source algorithms like legal line algorithm, similar to pharmacophore in commercial packages. So there are catalyst pharmacophore algorithm in the Discovery Studio. So actually there are a lot of algorithms that can be processed and can be used. And sometimes they're also very helpful. Some pharmacophore models are simple, they integrate only some activity centers in the molecule, and the other are more complex. They are also enriched by the molecular fields. So kind of a enriched pharmacophore concept. Some people even go in further. So here's an example of where enriched pharmacophore crescent fields. It's free for academia, but I also have this software, but I didn't really test it. But it's possible to test. And you could actually implement it in a rational drug design. So you can see you have also pi stacking here, different pi stacking ion fields, indicated strong interaction. You can have kind of stickier surface shape fields, hydrophobic fields, positive, also positive hydrogen bond donors, negative hydrogen bonds. So, and some negative ionic fields, aromatic pi hydrogen bond donor, small field showing less strong interest. So you can analyze this less strong interaction, very strong interaction, something in between. And actually the idea is to kind of simulate the protein ligand binding sites with this kind of pharmacophore. Usually you don't really have the actual representation of the binding site, but you can just extract this information by implementing these molecular fields. Sometimes people integrated these molecular fields in a quantitative structure activity relationship, like comparative molecular field analysis, similarity index analysis, confined CONSIA. And this is also sometimes very powerful tool in implemented in chemoinformatics algorithms. Okay, so the second would be the protein ligand interaction. So I would probably give more information on the protein ligand docking. We can probably go dive into this in the next